Okay, so this video is all about mutations, and it is assumed that if you're watching this video, you've either already watched the video on protein synthesis or have already heard the lecture about protein synthesis in class, because if you haven't, this will really not make any sense. So first of all, we need to define what a mutation is. So a mutation is any change in a DNA sequence. Now, if you have a change in a DNA sequence, it will, of course, result in a change in the RNA sequence when transcription occurs, and therefore, if the RNA is altered, then the amino acid sequence in the resulting protein will, of course, also be altered. But a mutation is only a change in the DNA sequence. So the first thing that can cause a mutation in a DNA sequence that we're going to talk about is an insertion. An insertion is when you end up with an extra base. And before we really talk about what would happen if this happens, I want to talk about what a triplet is. So a DNA triplet is a group of 3 bases that when it's transcribed actually would be the equivalent of the codon. So you already should know what a codon is. The DNA equivalent of a codon is a triplet. So if you add an extra nitrogen base, it shifts all of your triplets over, which would shift all of your codons over after the insertion. A deletion is just like it sounds. It's when you lose a nitrogen base. And this, of course, also shifts the reading frame of your triplets over by 1. So both insertions and deletions result in what's called a frameshift mutation. And so it's like a shift in the reading frame. So every single codon that would come after that part or that mutation in the gene that codes for that mRNA would be changed. So it's important to remember that with a frameshift mutation, you're always going to have a change in the triplets, and then you're going to have a change in the mRNA molecule after transcription, and then you're also going to have a change in all of the amino acids that are coded for after that insertion or deletion. So not only does it always result in a change in the protein, but it's always changes many amino acids in the protein. So it is a big change in the protein. And protein, if you're missing a protein or your protein is completely the wrong shape, it's going to cause major problems for that cell. A point mutation is a change or an exchange between one single nucleotide for another. This can be anywhere from no big deal to a really big catastrophe. So it can either change one amino acid, change no amino acids, or perhaps cause a premature stop. So you see right here, there are some codons that tell the cell, hey, stop here. So you could certainly get a very abbreviated or short protein that's completely non-functional, but that's not very common. So let me give you one example. So with sickle-cell anemia, the normal DNA triplet would be CTC, and the T is replaced by an A, you can see right here. So when we make our mRNA codon, or when transcription occurs, you would end up with GAG, because C is complementary to G, T is complementary to A, C is complementary to G, and instead you would have GUG. So now, if you go over to our codon chart and you go to G, from GAG, you go from glutamic acid to GUG, so GUG would be valine. So there is one amino acid difference. Now, there is a disease called sickle-cell anemia in which there is one amino acid difference, and valine has replaced glutamic acid, and the 3-dimensional shape of the protein is altered enough that it causes symptoms, but it is survivable. So people with sickle cell anemia often end up with joint pain, and it actually changes the shape of the cell. And it can cause problems, but it is not necessarily fatal, although it can be under the wrong conditions. I was about to say under the right conditions, but now let's look at another scenario. What if CTC were replaced by CTT? So then our mRNA would go from GAG to GAA. And then if you look at the codon chart, you go GAG, right? And GAA, they're right next to each other. And in both cases, both of those codons code for the same amino acid. So here you have no change whatsoever. And this is called a silent mutation. So point mutations can be silent mutations and not cause any change in the protein. That can't happen for a frameshift mutation. One mistake that students make when we ask them what is the real difference between the two, they say, oh, well, a frameshift mutation always changes the protein, and a point mutation sometimes doesn't. Sometimes you have a silent mutation. And while that's not wrong, you're also missing the idea that if there is a change, it's typically a smaller change with a point mutation, changes one amino acid, versus in a frameshift mutation, it would change every amino acid typically after that insertion or deletion. So it changes many of the amino acids instead of one. Now, of course, you can have a premature stop with a point mutation. So one interesting mutation is cystic fibrosis. About 70% of people with cystic fibrosis have a mutation in a protein called the cystic fibrosis transmembrane receptor, and it results from a deletion of 3 bases. Now, the interesting thing with this is that the 3 bases that are deleted are an exact triplet. If it had deleted 2 bases or 1 basis, it actually would have been worse and caused a frameshift mutation. Or if it was caused by deletion of 1 base in one triplet and 2 bases in another adjoining triplet, it would also have caused a frameshift mutation. But because an entire triplet is lost, you're missing an amino acid. So a phenylalanine, which is a kind of amino acid, is missing at the 508 position. That's like the 508th amino acid in the chain. So it is survivable to an extent. Frameshift mutations, you rarely hear about diseases that are caused by frameshift mutations because they wouldn't be survivable. And we only really hear about genetic diseases if somebody survives and has them, but has some sort of symptoms that we, try and figure out the cause of. Now, the next couple slides, you may or may not have spaces for in your notes. They're not in there this year. That this in 2019, because I just added these slides today. But it's possible that in future years I may actually add spaces into the note packet. So if you're watching this and it's not 2019, you might actually have space for this in your packet. But the reason I wanted to talk about some common misconceptions is that I've noticed over the last several years, that there are some common errors that students make on upcoming projects or lab analysis questions. So the first common misconception about mutations is that it's a mistake made during transcription or translation. The deal is that there are definitely mistakes made sometimes during transcription or translation, but when a cell is making protein, it's not making one copy of a protein. If a beta pancreatic cell is making insulin, it's making probably thousands of insulin molecules. So if you have a mistake in transcription or translation, that would mean one of those insulin molecules would be all messed up. But if the rest of them are fine, you're still going to have plenty of insulin in the body. The real problem is if there is a mistake in the original DNA that codes for the insulin protein. So in the insulin gene itself, that would mean that the beta pancreatic cell itself couldn't make any normal insulin proteins. So mutations are really only a problem. We only really worry about mistakes in the original gene and therefore the DNA. So this means that it is a mistake during DNA replication that causes mutations, not transcription or translation. The second misconception that I want to talk about is that all mutations are passed down to offspring. Mutations that happen in a body cell, like in your toe or a cheek cell or in your skin, can't get passed down. Only when a mutation happens during replication in a primary spermatocyte or primary oocyte, which are cells that are going to become eggs or sperm, can you end up with a mutation that gets passed down to the next generation. So most mutations don't get inherited by your offspring. The next common misconception is that all mutations are caused by the environment. While there's definitely are environmental factors that can cause DNA to mutate, like ultraviolet light or certain chemicals, sometimes mutations just happen by random mistakes made during DNA replication that the proofreaders don't find. Anytime you're copying DNA, there's the chance that it's going to go wrong, and it may not have any cause whatsoever. And the last misconception that I want to talk about is that all mutations cause cancer. We talked about cancer in a previous lecture and told you that all cancer is caused by mutations. But not all mutations cause cancer. Remember that in order to get cancer, you'd have to have a mutation in a gene that controls the cell cycle. So for example, a mutation in the gene that makes hemoglobin is not going to cause cancer. It might cause sickle-cell anemia, but not all mutations are going to be in cell cycle controlling protein genes. The last one that I really want to talk about, and I didn't write this on the slide, is the idea that all mutations are bad. Certainly, a lot of mutations are bad. Some of them are totally neutral, and some of them are actually helpful. Any new trait in a species has come from a mutation. And sometimes, if a mutation is helpful, it will get passed on in the species by natural selection and actually increase in the population. So, yeah, a lot of mutations are, bad, and some of them are deadly, but some of them are actually even helpful.